Welcome students. So this is the video that I am making on the topic of cell biology. So first thing is that this concept of cell that we have been studying for many years in every classes almost we have been studying about the cell, but this video is specially made for the 11th standard students. Okay, so the topic that I have said is cell biology. Okay, so let's start with the class, the video now. But before that, I just want to say that please sit with your copy and pen so that you can note down all the important informations, including the key terms, okay, the diagrams, everything. Okay. So let's start. First of all, I am going to discuss a few scientists who have contribution in the field of cell biology. Okay. So these are popular names. First is Robert Hooke. Obviously, he was the person who first discovered cell, okay, in the cork, and he was the person who first coined the term cell. Okay, so cork tissues are found in the bark of the plants. Okay, the second person is Anton von Leeuwenhoek. Okay, he was the person who first discovered a living cell. A living cell. Okay, the tissues, the cells found here are dead cells. Okay, so first living cell was observed or discovered, or you can say studied, by Leeuwenhoek. Okay, the third one, Robert Brown. He discovered nucleus. So he was the first person who studied the nucleus of the cell, Robert Brown. Okay, so now few more scientists. Schwann and Sladen. Okay, Schwann and Sladen, they first came up with the idea that all the living organism is made up of cell. So cell is the basic building blocks of all the living organism. Okay, this concept was first given by these persons. Okay, so they say that cell is the basic, structural and functional unit of living organism. So organisms, in short I am writing, organisms are made up of cells. Okay, but Schwann and Sladen, they did not talk anything about the generation of new cells, how the new cells are coming up. This portion was not told by Schwann and Sladen. So later on, Rudolf Virchow, he was the person who actually gave a final shape to the cell theory. Okay, that he was the person who said that how new cells are coming up. He was the first person who spoke about the cell division. That he said that new cells are generated from the pre-existing cell. So it means that it is the cell division that is giving rise to the new cells from the pre-existing cells. Okay, so pre-existing cells to the new cells formation. And then they sum up the cell theory. Okay, so 2 points were included. First, the organisms are made up of cells. The basic building blocks are the cells. Okay, the second point, that new cells or cells are getting originated or generated from the pre-existing cell by the process of cell division. Okay, so these 2 points that comes under the cell theory. So you can include these 2 points in the cell theory. Okay, fine. Our next topic I am moving now. Okay, few important topics that I have selected for today. The second thing is that we are going to see prokaryotes and eukaryotes. See, these terms all of us aware about this. Okay, I am just trying to recapitulate all the informations, where I am trying to sum up all the informations, because these are the basic informations required to understand the concepts that we are going to do in our next classes. Okay, so prokaryotes and eukaryotes, we are dividing the living organism into 2 categories, prokaryotes and eukaryotes. Okay, so what are the first basic difference between them? The prokaryotes are obviously unicellular. They are unicellular. Okay, while the eukaryotes are unicellular and multicellular both. Okay, first basic difference between them. The second one, you can say that in eukaryotes, okay, in eukaryotes we see a well defined nucleus. A well defined nucleus is seen in the eukaryotic cells, but prokaryotic cell, they don't have any well defined nucleus, so no well defined nucleus. Okay, now we have to understand what is the meaning of this well defined nucleus. It means the nuclear material, or you can say the nuclear plasm, including the genetic material, everything is surrounded by a nuclear membrane. Okay, so it's a nuclear membrane, okay, where the nuclear materials are present, including the nucleoplasm. Okay, so actually what happens is that this nuclear membrane is giving it a definite shape. Okay, but in prokaryotes we don't see any nuclear membrane, so there is no nuclear membrane here in the prokaryotes. So if there is no nuclear membrane, it can be easily understood that the nucleoplasm is scattered in the cytoplasm. So it is scattered in the cytoplasm, so it does not have a definite shape, it doesn't have a proper shape. Okay, so this one we can say, another term we use here, nucleoid. Okay, in case of prokaryotes, this is not well defined nucleus in prokaryotes. I hope it is clear. Okay, fine. One very important feature of eukaryotic cells is that we see membrane bound organelles. We see membrane bound organelles in the cytoplasm of eukaryotic cell. See, cell is having a membrane, we know. Nucleus inside the cell also have a membrane, now we came to know. Apart from the nucleus, in the cytoplasm there are many cell organelles present, and there are organelles in the eukaryotic cell, they have their own membrane which makes them separate from the cytoplasm. Okay, for example, ER, that is endoplasmic reticulum, they have their own membrane. Okay, then Golgi body or Golgi complex, you can say, they have their own membrane. Mitochondria also have their own membrane, in short I am writing here. Okay, and even chloroplasts also have their own membrane, etc. There are many. Even lysosomes also have their own membrane. Okay, so eukaryotic cells have these membrane bound organelles, where these prokaryotic cells, they lack all these things. Okay, they don't have any membrane bound organelles. Okay, so there are the basic difference between prokaryotes and eukaryotes. You must note down all these things in the copy. These are going to help you. Okay. Now next, before getting into the detail of the cell, actually we are going to discuss plant cell and animal cells in this chapter particularly. So before getting into the details of the cells, we have to differentiate between plant cell and animal cells. Okay, I'm not writing it, just let me discuss with this. See, plant cell and animal cell, basic difference we know. Okay, still let's make a revision. Okay, plant cell have the cell wall as the outermost layer. Okay, in case of animal, it is the cell membrane, which is the outermost layer, or plasma membrane, same thing. Okay, plasma membrane or cell membrane is also present in the plant, but that is the second outer layer. Okay, after the cell wall, then we see cell membrane in the plant, where cell membrane is the outermost layer in case of animal tissues, animal cells. Okay, now one feature related to these membranes is that cell wall is freely permeable, so any substance can easily cross through the cell wall. Okay, while in case of cell membrane, this is semi-permeable. Okay, semi-permeable or selectively permeable in nature. Okay, then there are some other differences. Now if we get inside the layers, if we get in the cytoplasm now of both the cells, plant and animal, then what we see? We see some more differences, like for example, vacuoles, the very important organelle of the plant tissue, which is not found in the animal tissue. Okay, in some animals it is found, but it is also very smaller sizes. But in plant tissues, the vacuoles are of large size. For example, in the parenchyma tissues, parenchyma cells, we see large vacuoles. Okay, they store cell sap. Okay. Second thing is that centrioles. Centrioles are not found in the plant cells, they are found in the animal cells, which have an important role to play during the cell division. Okay. Third, we can say that chloroplast, okay, plastids, which are present in the plant cell, not in animal cell. So plastids have to play a very important role during photosynthesis and pigmentation of various parts of the plant, which is not there in the animal cells or animal tissues. Okay, so these are some of the basic differences between plant and animal cells. So you also may jot down these points. Okay, now the next we are going to discuss about the cell wall. Okay, cell wall means it's the outermost part of the outermost layer, membrane of the plant cell. Cell wall, first of all, we already came to know it is a freely permeable. Now, what are the components of the cell walls? Basically, these are the polysaccharides. Okay, polysaccharides are the complex carbohydrates. Okay, made up of many monosaccharides that gives a complicated structure that is called polysaccharide. So whenever polysaccharides are present, it is actually giving it a very structural support, mechanical support. Okay, so that is the polysaccharide property. So some polysaccharides found in the plant tissues are, for example, cellulose, you can say. Okay, we also see pectin, we also see lignin. Okay, these are etc. So these are some polysaccharides we see on the cell wall of the plant. Okay. Now what I am going to discuss next is very important. This till now whatever I discussed, okay, detailing of these topics are not much required at this stage. Okay. Now the next topic that I am going to discuss, we have to study this in details. Okay, the structural feature is very important of cell membrane or plasma membrane. So our next topic now I'm going to discuss is the cell membrane or plasma membrane. Okay, so before we get in the structure, we have to see there is a model. Okay, model proposed that described about the structure of the cell membrane or plasma membrane. This is known as lipid bilayer model. Okay, lipid bilayer model, or this is also known as fluid mosaic model. Okay, this model was proposed by Singer and Nicholson. Okay, in 1972 most probably. Okay, this model was proposed. Now from this terms we can have some idea about the cell membrane. Okay, lipid bilayer. So there are two layers of the lipids, fat molecules in the membrane. So that indicates that majority of the cell membrane is made up of the lipids or the fats. Okay, and fluid mosaic model, another name. Fluid means it can be understood that it is giving a fluid property to the membrane, fluidity. Mosaic is the structural, you can say feature. Okay, texture. It looks like a mosaic model. Okay, now one information is that it is very clear that cell membrane, if I see the constituents, most of the components are lipids. That is very clear. But RBC is a cell, okay, that is having the proportion, if I see, 52% proteins, 40% lipids, and rest we see the carbohydrates. Okay, now see, proteins are more than lipids. That is in case of RBC. This is very important informations. Okay, this question was asked twice or thrice in the NEET examinations, in the medical entrance examinations. Okay, so this is the basic informations, okay, of the plasma membrane. Now I am going to start with the detailed structure of the plasma membrane. You have to learn how to draw the diagram of the cell membrane. You have to practice this. Okay, labeling is also very important for you. Okay, so let's start with the diagram of the cell membrane. So first of all, we have to draw the lipid molecules which are present on the cell membrane. We have to draw two layers. As you have seen, it is a lipid bilayer model. So one layer at the outer side, another layer at the inside. So two surface, keeping a little gap here. If you can start drawing with me in your copy, it will help you to learn. Okay. These are all the lipid molecules I am drawing here. Okay. Again, let us keep some gap here. drawing the double membrane is very important. Okay, 2 layers you have to draw. Again, let us keep some gap here. These gaps I am going to fill with some other parts. Okay, that's why I am keeping the gaps. You have to level this while you are drawing the diagram. Okay, so now it is clearly understood that how the 2 layers are there. Okay, this is the upper layer and the lower layer of the lipid molecule. So let's level it right now. Only we have to understand a little bit about the lipids also now. Okay, so let's level it thus. These are the lipids, as we already know about this. Okay, now lipids, there are different types of lipids. These are basically the phospholipids, the phospholipids. Okay, now phospholipids again is a group of lipids. So specifically if I say these are the phosphoglycerides. Okay, some property you have to know about this. First of all, these phospholipids, okay, they have a hydrophilic head and the hydrophobic tail. Okay, so these are the heads here. These are the heads on both the surface. Towards the outside we see the heads. Okay, so I am just pointing them separately just to make you understand that these are the heads of the lipid molecules, the phospholipids, which are hydrophilic in nature. Okay, so these are in just small, I am just writing a hydrophilic property, hydrophilic heads, okay, and these areas are the tails. Inside you see these are the tails. Okay, so this part is the hydrophobic tails. These are hydrophobic tails. Okay, this is very important feature for the phospholipid particles. Okay, now from this property we can easily understand that it gives the cell membrane a property of semi-permeability. Okay, that all the components are not going to pass through, as this area is hydrophobic, so water soluble components, they cannot pass through this area. So that makes it a semi-permeable condition. Okay, membrane. Now these phosphoglycerides, so these are phospholipids, specifically phosphoglycerides. Okay, they have one more property, okay, that they are liquid in nature. So these are actually liquid in nature. So as they are liquid in nature, so it can be understood from this that they give this fluid nature to the membrane. So the property of fluidity that comes because of these phosphoglycerides, these lipid molecules in the plasma membrane. So this fluidity also helps there are proteins present in the membrane that we are going to see. These proteins can easily they move laterally through the membrane because of this fluidity nature of the plasma membrane. Okay, now let me, so these are the lipid part I am done with. Now I am going to draw the proteins. Okay, see, there are two types of proteins found in the plasma membrane. Okay, integral proteins and peripheral proteins. Integral protein means the proteins which are integrated or embedded inside the plasma membrane. There are 2-3 different types of integral proteins I am going to draw. This is the first integral protein. Okay, now you can see there is a channel in between this structure, a channel or tunnel like root we can see here. Okay, So the functioning of this membrane definitely we are going to discuss separate topic. Just for now, to get a little idea about this, these are the proteins that helps in the transmission of those molecules, okay, which cannot directly pass through the lipid particles, lipid molecules. Okay, these proteins help them to transport or move across the membrane. Okay, so this is one of those integral protein. So the name of this protein is channel protein. As we see, the channel here, the channel protein, or you can also say tunnel protein, the same thing. Okay, now the next is this is another type of protein here, integral protein again. Now this one you can say is a globular protein, globular proteins. Okay, another one, let me rub the upper portion from here. Okay, this is another protein which is known as alpha helix protein. Okay, so these proteins, the function, as I have told you, that they helps in transmission of the substances, okay, or helps in move across the cell membrane. Okay, these proteins, they function as a carrier or transporter of those substances. So all these three are different types of integral proteins. So they are integrated or embedded inside the plasma membrane. Okay. So okay, this is integral. Now I am going to draw the peripheral proteins which are also present there. So these are the peripheral proteins. Peripheral means, the term came from the periphery, means the outer membrane. So these proteins are found towards the periphery of the membrane. Okay, so these are the few proteins I have drawn here. So these are the peripheral proteins. Okay, function remains same. These proteins, because of the fluidity nature of the plasma membrane, they can move across laterally through the plasma membrane, and they can help in transportation of the substances, okay, throughout the membrane. Apart from that, what we are left with? We are left with the carbohydrates now. Okay, one very good information about the peripheral proteins are that these peripheral proteins, okay, they are with this, you can say hydrophilic heads. They are in attachment with the hydrophilic heads with the help of hydrogen bonds and ionic bonds. Okay, this is very important information here. So they are at the surface area, so they are in attachment. They binds with the head portion of the lipid particles with the hydrogen bonds and ionic bonds. The tails here inside, okay, there is no chemical bond here. One that I was going to discuss about this, that these tails of these lipid particles, these are actually, they binds with each other by van der Waals force. Okay, I hope we study about these things, or we are going to I'll be discussing about this van der Waals force also later on. Okay, so inside these tails are binds with each other, okay, by the van der Waals force, and outside the hydrogen bonds we can see our ionic bonds. Okay, now we're left with the carbohydrates. So let's see the carbohydrates here. I'm using a separate color for this. So these are the different types of carbohydrates here. Okay, so 4, you can see that I have drawn here intentionally the 4 carbohydrates, not more than that. There are many, but I have drawn 4 because just to make you understand something very important, that these carbohydrates, if you look very carefully, that 2 of them, this one and this one, these 2 of them, these carbohydrates, they are connected with the proteins. With one is the integral protein, one is the peripheral protein. So on the basis of this connectivity to the proteins, we are naming them as glycoproteins. So now I hope it will be easier for you to remember the names. Okay, glycoproteins are the carbohydrates which are in attachment with the proteins. And now there are 2 carbohydrates you can see here. They are in contact with the lipid molecules. Okay, so these are known as, I'm writing here this one, these are glycolipids. Glycolipids. Okay, now what is the function of these carbohydrates? Cell to cell recognition mechanism. In this function they play a very important role actually. Okay, cell to cell recognition mechanism is something like a good example for this is fertilization. The concept is that when a foreign cell or foreign germ, in case of that any cell, if they enter into our body, okay, then my cells, okay, they have to identify whether the foreign tissue, the foreign cell is our enemy or our friend. To recognize them, the mechanism that comes in to play important role here, cell to cell membrane recognition or recognition mechanism. Okay, one good example, as I said, is the fertilization. What happens is that when the sperm enters into the body of the human being, then it is going to fuse with the egg cell. We know this is the fertilization, that the male gamete fuses with the female gamete. Now what happens is that they are fused, the sperms get fused with the egg cell. So both are cells. The sperm that is coming, that is the foreign cell coming outside of the body, it is going to fuse with the egg that is inside the body already. So how to recognize them, whether this is the exact cell that the egg cell want to get fused or it is something else. Okay, to recognize them and then allow them to get inside is the function of these carbohydrates. Okay, so in short I have told you this cell to cell recognition mechanism. There are some more to tell about it. When the topic will come, I will definitely discuss about this. Okay, so let me see there anything left here. Okay, so now you can see this is the diagram. Almost I am done with this topic for today. You see these proteins they are unevenly scattered in throughout the plasma membrane. It looks like the icebergs in a sea of phospholipids, right? The whole phospholipids are there and they are like icebergs in the sea of phospholipids. So they are unevenly distributed throughout the plasma membrane. Okay, and helping in the transportation or helps the molecules to move across the membrane. Okay, so this is it for today. In my next topic, I am going to discuss with the detail of the transportation mechanism throughout this plasma membrane. Okay, let me see if anything left. Just before ending, okay, one more additional information I am going to give. Okay, this you see something that I am drawing here. They are also found at the head side of the phospholipids here. These are nothing but cholesterol. Okay, so I am labeling here. These are nothing but cholesterol. See, these phospholipids, they are what? They are giving the fluid nature to the plasma membrane. But what these cholesterols are? The cholesterols are chemically rigid in nature. So they are providing the rigidity to the plasma membrane that helps the membrane to maintain the stability of the structure. Okay, so this is how, as the fluidity nature is also there due to the phosphoglycerides, the rigidity is also provided by the cholesterol. Okay, so the functioning is also going on, the stability of the structure is also maintained by the cholesterol. Okay, so this is it. Thank you so much. I hope it is going to help you a lot. Okay, so thank you.